Thank you everyone for joining in. Welcome to Axiom Think Tank. I'm pleased with my colleagues here in Princeton, as well as Rohan Bipani in our Boston hub. We're an oncology and rare disease focused think tank on addressing key issues as it relates to scientific research, as well as issues impacting our patients. With that, Rohan.
Thank you very much, Hafiz. I want to welcome everybody to our latest installment in the Axiom Town Hall Roundtable series. Today we'll be looking excitedly at the space of cell therapy. And while we have discussed autologous methods before, including marketed products and products seeking additional indications, today we're going to be taking a look at some of the newer technologies that are enabling the allogeneic techniques, as well as techniques that enable us to enter solid tumor cell therapy. And with us today, we have three esteemed key opinion leaders. We have Dr. David Hong from the Department of Investigational Cancer Therapy, Division of Cancer Medicine. We have Dr. Cassin Yi at the Department of Melanoma Medical Oncology, also Division of Cancer, both from the MD Anderson Cancer Center. And we also have Dr. Butler, who is the clinical head of the immune monitoring team at Princess Margaret Cancer Center, as well as assistant professor at the University of Toronto. So without further ado, I will turn it over to our esteemed panel.
So I don't know if Dr. Yi is on. Is Dr. Yi on? I think he's going to talk to us today about cell therapy as cancer treatment in general, and then I will go over some of the autologous cell therapy first steps and challenges. Then Dr. Butler will talk about allogeneic cell therapy, some of the issues that are ongoing there and progress to date. Then I'll end with cell therapy and solid tumors. And I think there'll be a number of questions in between each of those sessions, and then we'll end with a Q&A. Is Dr. Yi on?
Great. Can you see me or hear me, I hope?
Yep.
So I think I've got the responsibility of laying down the landscape for adoptive cell therapy in the context of immune-based therapies. We all know that there's been a renaissance, not just in the immune checkpoint field, but also in cell-based therapies. And as it's divided here arbitrarily, you can see on the right-hand side of the screen, basically non-engineered adoptive cell therapies versus engineered cell therapies. I'm not going to touch on the checkpoint inhibitors. I think we all know about that and how successful that has been for a number of cancers, initially melanoma, but also lung cancer, but now a whole broad swath of cancers that have infiltrating lymphocyte population, and then bispecific antibodies, a rendering of some of the technology that's been available for at least surface targets. The adoptive cell therapy addresses a question, which is if you don't have enough T cells, or if you don't have enough mutational burden in a lot of these tumors, perhaps you can supply these T cells. TIL's been around, as many of you know, for 20 years or so. Initially pioneered in C. Rosenberg's lab, but now elsewhere. And basically taking out the tumor, extracting the lymphocytes that are present, expanding that ex vivo, and then giving that back to patients in the context of lymphodepletion. The other non-engineered cell therapy is endogenous T cell therapy. Marcus and I have both worked in this field, but others as well, in which we isolate the T cells from the peripheral blood and then recapitulate the in vivo antigen-presenting cell T cell stimulation process ex vivo in a while. And then some means to isolate, expand, sort, or what have you, isolate these tumor-active T cells, expand them to large numbers, and then give them back to patients. And we can talk about the balance of these two later, but I think for the context of this discussion, we're going to focus on TIL therapy as a non-engineered ACT because there's a lot of industry interest in that and commercialization of this potential. And contrast that to engineered adoptive cell therapy, which is basically redirecting the specificity of a peripheral blood lymphocyte population by expressing either chimeric antigen receptor, which is the antibody attaches to surface antigen, or the T cell receptor. And in this case, we're talking about CAR T cells. Next slide. So I said I was going to focus just on TIL in terms of non-engineered T cell therapy, and this is what we're getting here. Basically, the process is isolating TILs from the tumor. I've mentioned some of this already. Turns out that a lot of these TIL cells are CD8 T cells, although there's a mix of other cells present that are expanded, ex vivo NK cells, some CD4 cells, and so on. But for a source of effector cells, it seems a lot of these TILs will have T cells that recognize either neoantigens or tumor-associated antigens and so on. And it's hoped that because you're taking it from the tumor site, they also traffic back to the tumor site after you expand them. And the ways that people are trying to refine this process a little bit is either enriching for CD8 T cells, enriching for activated cells, identifying a repertoire that may be more likely to be tumor reactive. The reinfusion process, just to keep in mind that this doesn't act alone, that you need some sort of lymphodepletion, usually a non-myeloablative regimen, or post-infusion treatment with high-dose IL-2. Again, these are potential barriers, but also being addressed and modified to try and streamline TIL therapy for the general population, even beyond melanoma. Next slide. So these are some of the enhancements I touched on in the previous slide, which is can you genetically engineer them to home to tumor sites like CXCR2? Can you also downregulate molecules like the TGF-beta dominant negative receptor, putting that in to undermine the process of immune suppressive cytokines that might be present in tumor AU. So these are all sort of engineering principles that may be first adopted in TIL, but also applied to CAR-T, TCR-T, and ETC therapy as well. Clinical trials have been done. The CXCR2, I guess, notably was done here at MD Anderson for a little while. And the combination with immune checkpoint inhibitors or TME immune agonists is something I'm just going to have to say right out front was started with ETC therapy because of the low toxicity, but TIL is coming on to that sort of bandwagon in a way that has to address certain issues related to tumor microenvironment. As I said earlier, you can give the immune checkpoint therapies, but no matter what you do in terms of taking the brakes off the car or the vehicle, if you don't have the vehicle, which is a T cell, nothing's going to happen. The use of allogeneic HLA matched TIL, presumably for off-the-shelf administration, is very early in the process, and I think that's something that we may touch on later in this discussion about the use of allogeneic cell products. The number of clinical vignettes, I'm not sure how much of this you really want me to go into, but I think it's fairly clear that the publication very early on, about 10 years ago, showed that TIL plus high-dose IL-2 was effective for a subset of patients in terms of response rates. Now, I have to caution here, I think this is the best overall response rate, 72%. That was in a very, very, I would have to say, had huge lead time bias in that study where where ablative radiation was given to patients. But in general, the response rate's around 40% or so, at least among the institutions here, Sheba Medical Center, MD Anderson, and Rosenberg. And 5-year survival can be as high as 90% if you have complete response. But I think those are the best figures. Seriously, when you're looking at this from a more general standpoint, in subsequent studies, this number is actually a little bit lower, although there's a subset of patients who have a complete response. The use of low-dose IL-2 until, we actually did that at Fred Hutchinson initially, and other people have done it now in Europe, and I think this is promising. Obviously, obvious requirement for intensive care unit to look after these patients after the use of high-dose. Jury's still out on this. I think whether low-dose IL-2 is equivalent to high-dose IL-2. There's certain benefits of high-dose IL-2, such as IL-15-like effect and trafficking and so on. Nevertheless, the toxicity is more overwhelmed the ability of potential benefits over low-dose IL-2. So I think this is certainly something that people should be considering in this context. And then finally, as you all know about the phase 2 advanced melanoma study, lifileucel in patients seem to have a pretty high disease control rate, 80% or so. I think, again, as with all these types of studies, keep in mind the lead time bias, the patient selection, and so on. I wouldn't hold these numbers hard and fast, but it's certainly promising for the field. Next slide. Oh, discussion time. I'm not sure if there were any questions for TIL related. We can talk about it now or a little bit later. I realize I only have 10 minutes to go through the next few slides.
I think we'll go pretty quickly. I'll just encourage the audience to ask any questions in the chat function, and we'll try to get to them at the end of the session. We have 30 minutes for Q&A. I'll just ask very briefly, in terms of, for TILs itself, are these only restricted to hot tumors, or is there any hope for, we talk about pancreatic cancer being pretty cold and inhospitable. Is there any hope for engineering methods that would allow success there?
Well, that's a tough question. I mean, I think there is some evidence that you can isolate TIL that are tumor reactive from what were considered cold tumors like pancreatic cancer. And you may need certain You're forcing me to talk about work that's being done here in MD Anderson, so I'm going to have to just say some of the studies that are published with anti-CD137 agonists, for example, does allow you to cull some of these out. I know the NIH is doing work with PD-1 positive selection, enriching that reactive population. So even though there's a small number of what you consider cold tumors, small numbers of T cells, it seems you're able to isolate these tumor reactive cells in the population. I think that it's still a high barrier, and which is why TIL may not be applicable for all types of tumors.
Super. Thank you very much, Dr. Yu. We'll jump into your next section. Sorry.
Mm-hmm. Oh, yes. So this is interesting because I think this is something that is close to my heart. It's like, what is it that's happening in the natural process, a physiologic process? T cells possess an ability to recognize tumor targets, but what is it that stimulates these cells, and in what conditions, ex vivo or in vivo, and how is this message passed from the antigen presenting cell, in this case the dendritic cell, to the CD8 T cell through its T cell receptor? Next slide. And so I guess this is sort of the basic principles, is that antigen is processed and presented either by tumor itself directly or through a professional antigen presenting cell, in this case, a dendritic cell. And so here we're taking an example of the dendritic cell that's collecting, processing, and presenting small fragments of the protein, a tumor-associated antigen, tumor-associated protein on the surface as epitopes of peptide. And they must be presented on the surface with the self-MHC molecule, which is the HLA type, and of course, the HLA allele. And then this interaction with the T cell receptor is really the first signal. There's other interactions that are required, not shown in the slide, for example, but costimulatory signals, accessory signals to increase binding, allow the T cell to reach a threshold of activation, which then releases perforin granzyme, punches holes in the tumor, and then affects tumor killing. They also release certain cytokines or effector or chemokine molecules to attract other effector cells to the site to order expand or multiply or exploit the whole inflammatory response of a T cell arriving first into the tumor site. I think the one important thing is that when you're doing a T cell, looking at T cell responses, you can, that T cell response recognizes an epitope that can come from any part of the protein in the tumor cell, not just on the surface. It can be tumors that are broken down, transcription factor, nuclear factors, and so on. So there's a much wider array of tumor targets if you're using a T cell receptor. Next slide. Oh my goodness. Okay. Yeah, I knew this was going to get complicated. So let me just say, this is the antigen processing and presenting pathway. I'm not sure how much you want me to go into the detail, but there are two different pathways. On the left, you can see class 1 pathway, which requires a tap to transport these endogenous proteins, usually from the endosomal reticulum, and then through a proteasome where parts of the peptide, which is maybe too long, is cleaved to fit into the MHC and brought onto the surface. And the same for class 2 is processing does need to take place, but through different organelles within the tumor antigen presenting cell, and then brought to the surface in the context of class 2. Class 2 is usually associated with exogenous antigen that's taken up, although there is a pathway for exogenous antigen to be presented through class 1 as well. So the rules are not hard and fast, but the most important rule here is that it must be presented in the context of a self-MHC class 1 for CD8, MHC class 2 for CD4. Just go to the next slide. I don't want to take too much time away from the other speakers, but I think that what CAR does is it sort of redirects the specificity. Now, the two types of engineered cell I mentioned before, CAR, chimeric antibody receptor, which is an antibody that recognizes surface, and TCR, which is a T cell receptor recognizes peptide C. With CAR, you're taking the surface recognition molecule, in this case, an antibody, chimeric antibody, and then you're signaling through the same sort of apparatus that the T cell receptor normally signals through. So you're still within the T cell environment, but what triggers it now is the chimeric antibody receptor recognizes surface antigens. And this is what's been done by Michel Sadelain and Carl June and others when they see the CD19 on the surface of B cells. It does say here that one of the important things is to knock out the T cell beta or track locus. I think that's more important for a T cell receptor so that there's not mispairing them for a CAR. But for a CAR, I think sometimes you want to put the CAR within the track locus so that cycling of expression of that CAR occurs more I guess physiologically. You don't want it on the surface too long, or you may end up with activation-induced apoptosis, exhaustion, or what have you. So that's something actually I think Michelle Sadelain had developed, and certainly being used by many other companies and industries for the sort of naturally CAR surface expression. Next slide. Is this still my section, or?
No, it's my section.
Go ahead. Thank you. Yeah, all right, I'll pass it on to David now.
All right, let's go. So, it's interesting. I remember doing not this kind of session, but it was in a kind of a advisory session. I don't know, almost 10 years ago, and I remember cars were just coming out, and somebody asked me in that group session what I thought the coolest next kind of platform of therapies was, and I mentioned cellular therapy and CAR T cells, and whoever the analyst or whatever in that group, I think it was a guide point thing, laughed and said that will never happen. That will never happen. It's too complicated, and there's too many things in order to get these things up and running. I hope that guy does not have a job anymore, because clearly he was wrong. And we are now, as Cassian mentioned here, already here with multiple approvals, obviously Kymriah, Yescarta, Tecartus, all of these now are in the clinic, being utilized in patients, particularly with obviously diffuse large B-cell lymphoma, but other hemolynicities and ALL and other things that will eventually emerge. Next slide, please. But one of the things I think we've started to realize is that this therapy is not your standard antibody or small molecule or even chemotherapy. We started here at MD Anderson many years ago something called the CAR-Tox group, because as we were running some of these CAR T-cell patient trials, one of the patients on the study had died of ICANS, which is this neurotoxicity. We recognize these therapies are not benign. They can have significant toxicities, CRS, ICANS, that could even lead to, in some instances, death. Beyond that, there are other major challenges to autologous therapy and just cellular therapy in general. Biologically, we know that antigen escape can happen, and just allowing for the persistence of these cells is also a significant challenge, and particularly in solid tumors, traffic infiltration. Dr. Butler and I run many of the solid tumor trials in cellular therapy, and there are lots of logistical challenges, whether it's from leukapheresis, manufacturing, the actual distribution and cold chain of custody, and then following these patients post-reinfusion. Beyond that, one of the things that, obviously is still a challenge, despite the fact that these drugs or these platforms are approved, is cost. We still get significant pushback from insurers in making sure that these patients can actually get some of these therapies. On top of all this, particularly in the context of clinical trials, there are additional layers of complexity, whether it is, different dose levels, trying to navigate which patients from multiple centers can be enrolled in a dose cohort, and on and on and on. And so these CAR T challenges and logistical challenges are not minor. Next slide, please.
So for this discussion, I want to open it up to the panel. The first point I want to emphasize, or perhaps ask about, is, is there some aspect of patient factors that features into some of the logistical concerns, maybe even biological?
Well, I think one thing that, just from a clinical trial standpoint, and I'll let Dr. Butler also comment on this, is, most of the trials right now that are in cellular therapy, for example, are late stage. These patients are late stage, right? So in solid tumors, for example, most of the patients that I see that we're trying to get onto these clinical trials have already failed standard chemotherapy and or standard therapy. And usually, even though their ECOG may be pretty good, their disease is, on a march. And trying to get them, not only screened, but leukapheresed, and then trying to somehow, keep them going to a point where that they can actually get the therapies within, usually it takes about 2 months, usually from the time, vein to vein time, still takes 6 to 8 weeks with a lot of these new therapies, and trying to maintain these patients in a point where they can actually tolerate these therapies sometimes can be a challenge. So that population, at least clinical trial population, early studies can be a challenge. I don't know what other in general in cellular therapy, probably the best predictive, marker, not to say markers, but characteristics of a patient is disease volume, right? So patients who, in general, in the patients who are getting in lymphoma, patients who have lower disease volume tend to do better than those who have high disease burden.
I think there are also factors that have to do with the starting immune cell population for patients that have been beat up with many lines of prior therapies. So in the current environment with the approved drugs, approved CAR T products, which essentially are established approval on the basis that these are the patients on their last legs, where a significant response rate with a high proportion of patients with durable responses means that you have an approval. But by definition, these patients have had auto transplants, they've had a few lines of therapy, and if the insurance company is only going to approve for that patient population, there's a high percentage of patients who generate a T cell product that's not really optimal because they've been so beat up after years of prior therapy. So I think in terms of unmet needs for the approved CAR T products, it's to figure out when is the appropriate time to infuse people early, perhaps in their treatment course. Maybe we'd have better durable responses. The cost is an inhibitor for doing that, but it's conceivable that we would have patient factors are influencing the quality of the T cell product, and we certainly know that exhausted T cell product doesn't allow for persistence, and therefore we need to figure out ways to bring this therapy forward. I don't know if Caspian has a lot of experience generating T cells from beat up patients, so you could actually comment.
So thanks, Marcus. I meant that in a nice way, actually. But I think that 2 points I want to make. One is that when we've treated patients, for example, with the WT1 specific T cells, and the patients who had minimal residual disease but high risk of AML, all those patients were cured as long as the T cells persisted. So to the point of what David raised, lower disease burden is important. Obviously, that's not the patients we get or anyone gets, in these cutting edge studies. The second point, actually, maybe not so well known, but maybe in the lymphoma CAR T cell field, we've done an analysis using pharmacometrics to look at the dosing and the patients and everything else. But this has also been previously published that patients who had an autologous transplant before collecting for CAR T cell therapy actually have a higher response rate, durable response, which is, to Marcus's point, that you have a fresh influx of cells in these patients that may provide, endogenously a product that has a better immune function or status or starting point anyway. But a great area of discussion.
Fantastic. I think that's a perfect segue into Dr. Butler's introduction into allogeneic overall.
So one of the big challenges with the autologous products is, as David was saying, there's a long lead time to generating sample. What you see in the lower left is patients having pheresis and then engineering the product for then harvest and infusion. The approved CAR Ts are shortening that period. And that's quite critical for getting patients treated. But it is on trials and in new biotechs, it is a big challenge. So an allo approach is where you use a donor to start with your T cell product, activate the cells, engineer the cells, typically using lentivirus or retrovirus, and then expand the T cells, harvest and infuse. But this product can be frozen and will essentially be on the shelf and ready to use. You can use various sources. You can identify the best donor population for generating cells for infusion. Next slide. Essentially, it's the logistics on the left in terms of leukapheresis and manufacturing that you're able to fix, and therefore you can identify patients and then treat them as soon as they're ready for infusion when you have a third-party donor for generating the cells. Next slide. So there are multiple approaches for generating an allogeneic product. As one of the challenges for a T cell is that's an allo product. When you infuse it into a patient, there is the potential for rejection of the graft, so that essentially the anti-tumor effect isn't able to kick in because the T cells are rejected either by an allo response or a natural killer cells. So a couple of techniques are used. One is using CRISPR technology to edit the donor T cell. You can not only put in the CAR T, but you can also knock out beta-2M, so you have class 1. It's not expressed. You can engineer the cells to express HLA-E so that that will reduce NK cell killing. You can knock out T cell receptor. You can also express CD52 as a way to for lymphodepletion and to allow the cell product to work. The other, not only are the engineered products currently being investigated, but there are also virus-specific T cell products that are being tested. Atara, for instance, is generating products with that are partially HLA matched to recipients, and then the allo T cells that are enriched for virus-specific cells can be infused as a therapy. Next slide. This is also a slide to point out that several groups are looking at natural killer cells generated from an allo donor and then used as a T cell product. You could either infuse NK cells that are unmanipulated, unengineered, or you can generate a CAR T-like product that is a natural killer cell and redirect that NK cell towards a tumor with a chimeric antigen receptor. Next slide. So the unmodified NK cells versus CAR T provide various advantages. In terms of targeting, if a tumor is sensitive to NK cell killing, then that's a straightforward, favorable effect. Whereas CAR T is engineered to go for a particular product. The unmodified NKs are usually rejected relatively fast, so requires multiple infusions. The target repertoire for an NK cell is broad, whereas a CAR T is specific for particular targets. So you may have less broad repertoire, but you have more tumor specificity. And there's when you generate a product that has a high chance of being rejected, then there's less GVHD risk. But then when you have a product that has higher potential for persistence, when you're using an allo product, especially if it's a T cell product that continues to have a TCR expression, you may have a risk of GVHD. Next slide. In terms of other concepts that are being investigated to engineer products, the CD38 knockout can provide resistance. IL-15 receptor engineered products can be generated so that you have a product that will have better persistence and effect. These products can also have TCRs specific for viruses engineered for infusion. Next slide. So this is a slide to note that there are several groups that are investigating stem cells for generation of an allo product. The advantage here is that if you're basically you have control over all of the aspects of the T cell product that you're generating, if you're basically starting from scratch and engineering those cells specifically for properties of interest, versus starting with a donor and generating T cells from that specific donor. iPSCs have the advantage that if you do come up with the right process, then you have an immortalized product that can continue to be generated over time. You don't have the issue where a particular donor works really well for generating your allo product, but then if that human donor moves away and is not able to provide more cells, then you may have variability from donor to donor. So I think it's certainly the holy grail to start with iPSCs, but we don't know yet what will be the best and shortest route to effective therapies that'll be approved. Next slide.
Great. So I want to open up discussion again, and perhaps we'll index on that last point around iPSCs. It seems to be a theme that's emerging. Everybody's talking about the benefits that they imbue, particularly for the allogeneic cell therapies. Is this something that is sort of too soon for making a judgment about? Are there manufacturing challenges that are both favorable but also might hinder iPSCs as an approach to take, given we already know about other manufacturing approaches? So just want to get a sense of where iPSCs are at this point in time.
I can start. I think it is too soon. It's a little too soon to make a call as to, whether which platform, which Marcus kind of highlighted and reviewed, is going to ultimately be, the winner in all of this. I do think that overall cellular therapy, in my opinion, this is my opinion, and overall that we have to, in a context of cellular therapy, move towards allogeneic, because I think that's the only way that we're going to scale this. I think the vast majority, and I think I'm correct on this, but the CAR Ts for now that are being given are given in primarily here in the United States. I don't know if it's the same true in Canada. My guess it is being given at academic centers like MD Anderson that have the resources to, admit these patients, have specialized teams to monitor their toxicities, give the leukapheresis, lymphodepletion. I think the same thing is going to apply towards TCR therapy. There's a good chance that, Adaptimmune is going to get their synovial sarcoma approval and their SPEAR T cells in their phase 2, probably sometime next year. But I don't see it being given anywhere other than academic centers, because I just don't see how you can put all of the bells and whistles and logistics laid out for a community, a doc in Lubbock, Texas. Like you have to be able to give this off the shelf in a way that doesn't have a huge amount of toxicity. And the only way to scale cellular therapy will be off the shelf.
Great, and perhaps as a second discussion point, I was wondering whether there are other, you mentioned bells and whistles. Are there any other bells and whistles or innovative approaches being implemented in allogeneic cell therapy, sort of thinking about gating mechanisms? SentiBio, I believe, has something there, or enhanced stimulatory domains. Are there any other allogeneic bells and whistles, as it were, that really come to mind in terms of interesting approaches?
I'll just take a stab at that first, Rohan, but I'm not sure if you're asking specifically to allogeneic products, because I think when you talk about bells and whistles and SentiBio and other people, it's a synthetic biology sort of concept, as well as sort of a TME specific sort of trigger for whatever it is, whether it's prevention of exhaustion, whether it's to obviate the effects of hypoxia, or whether it's, to increase proliferation at desired sites. So I can only imagine how that might apply more specifically to allogeneic products if you're concerned about that product lodging elsewhere or being rejected or killed off by its endogenous response before it gets to the tumor site. So a lot of the bells and whistles are coming from epigenetics and synthetic biology right now. I can't recall offhand if there is a specific genetic engineering other than what's been discussed already that applies specifically to allogeneic, but I'll defer to the other two discussants here.
Yeah, I would agree. I think conceptually the allo versus auto, the auto product has all the problems with manufacturing, but you have a lot of proof of concept, and then you don't have to worry about rejection as much. So if you have an allo product that is going to solve the problem of rejection of the product, then that's interesting. But in terms of bells and whistles that make an allo product traffic better or resist the hostile tumor microenvironment, I think those same lessons can be learned from the autologous products as well. And both of these strategies can learn from each other.
Fantastic. And with that, we'll move on to the allogeneic cell therapy section, where we'll discuss some of the data to date that has been published to date.
Is this Dr. Hong or myself?
I think that's you, Marcus, still.
Okay. So in terms of autologous CD19 CAR Ts, on the left you see the response rates and the 6-month CR rates published. And then on the right you see allogeneic CD19 CARs with recent data looking at quite high response rates, CR rates, and as well as 6 months. All of these studies are performed quite differently. The cross-trial comparison should be taken extremely cautiously. And clinically, we don't really look at these numbers and say, oh yeah, we really need one particular drug over another, because the patient populations are way too different. The conditions for doing the trials are too different. Next slide. One of the major concerns with the CRISPR technology is the potential for gene editing gone awry. So I'm sure this audience is well aware that Allogene has had to put all of their clinical trials on clinical hold due to the development of chromosomal rearrangements that are observed in patients treated on these studies that utilize TALEN. So this, the patient in question had a cytopenia that was prolonged. This is very common in the patients receiving CAR T or TCR T cell therapy seen in both heme and solid tumor malignancies, but there is that additional concern of inducing pathological genetic changes with this technology. So, one of the challenges I think will happen over time is that, or one of the challenges for the allo products is that there are additional safety concerns, especially for iPSCs, for instance, where we're going to have to follow patients for a long period of time to make sure untoward effects don't occur. So, in the sort of the counterpoint to Dr. Hong's point about allo being needed for off-the-shelf and more widespread use is that the safety concerns may be higher with these allo products, and therefore that might delay coming to market as patients are monitored. So next slide.
So we head into this discussion, I sort of want to circle back on that point about Allogene to the extent that we can. Of course, it's been a pretty recent observation. And given that it was the TALEN gene editing platform that was used, and also the fact that the company has disclosed that it was due to T cell expansion, and they state that it isn't related to the actual gene editing platform and that it would be broadly across allogeneic products. Where does that really leave us in terms of whether T cell expansion in vivo in general is going to be the problem across allo, or whether there's a gene platform specific issue?
So just before we get into this, I just want to be very clear. I haven't seen data, and I know that was still a question of whether in fact this was a gene editing issue or not, because it was in chromosome 14, and where specifically it was in chromosome 14 has not been disclosed as far as I know. So, the claim that, any expanding population is going to lead to mutations that can lead to a clonal expansion of a product that's, somehow has some proliferative advantage, I think is problematic from two standpoints. One is that, first of all, there's no evidence that this is the case in this product or other products, or why an allogeneic product would be more likely to do so than an autologous product. Any evidence for that. And secondly is that, I'm not sure if this is meant as a good thing or bad thing, but it does put a damper in the entire field if you're going to claim that expansion is an issue for an allogeneic product. And I don't think, I can't, intuit what the company's intention is by making such a statement, but I think it's a little bit hazardous, that's all. Yeah.
And then getting back to the actual data that was presented, taking the Cellular case, more recent from last week for CTX110, showing a poor 6-month complete response rate. Is there anything that we can read into that, given that it is 6 months, and we compare it to Allogene, it's worse than Allogene? But is it too early for us to consider 6-month CR as a metric? And if we do evaluate that over time, are we expecting a 12-month CR to be better or worse? How do we reconcile that sort of matrix?
To me, 6 months is not going to. I wouldn't bet my house mortgage on a 6-month CR rate. I think that it's a great thing to show when you can compare. The studies are done differently. A lot of times investors are looking at these short turnaround metrics, but I don't think it's not where the value is in these products. If the value of the product is what the disease control rate is a couple of years later. So sure, if the CR rate at 6 months is a true surrogate for that endpoint, then great. And that may be the case, but I think we need more data, and these studies are too small to do comparative. I don't know if Cassian feels differently, or David.
No, I feel the same way. I think a lot of the relapses in the original autologous CD19 studies occurred around that time, 6 months and later, and that was a high fraction of patients who did achieve complete response. And so, and now we're talking N of one or N of a few. So I fairly cautious as you are, and certainly without any other immunobiological data, it's hard to make a prediction based upon that. Yes, promising, just context.
Great. So with that, we'll head into Dr. Hong's section on solid tumors. And this is obviously a very interesting area, both in auto and allo. And so I'll just turn over to Dr. Hong to take us through that.
Okay, let's move on. Next slides. Okay, so the solid tumors really do present some unique challenges for cellular therapy, and I give credit to Cassian and all the initial pioneers who decided they were going to try to tackle this so many years ago. A number of things you can see here are some of the challenges, whether it's certain cell surface antigens or have low abundance, and heterogeneity is a problem. Antigen escape is a problem. Poor trafficking, I think this is also a major problem in the tumor microenvironment in solid tumors, and some tumors are worse than others. The actual immunosuppressive environment and T cell expansion and persistence, and again, TCRs can also have some low binding affinity to these HLA complexes. Next slide, please. So, Cassian went through this. This is a depiction of this, I think, from the adaptimmune spared T cells. It's really trying to reprogram the TCR to recognize specifically MHC-associated antigens. TCRs, some would argue, more so than even CAR, in the context of solid tumors, may be a better avenue or a better mechanism of targeting solid tumors. They seem to have high affinity. They seem to also activate, be more engaging the overall tumor microenvironment, whether it's with dendritic cells, et cetera, to really induce a robust attack on solid tumor cells. Next slide, please. So you can see here kind of the advantages and disadvantages of solid tumor therapy. In CAR T, CAR Ts really only target cell surface antigens. They don't target antigens that are processed endogenously and expressed on these HLA complexes. And the vast majority of antigens that are tumor specific, that you're not going to have widespread antigen expression on normal tissue, are expressed through these endogenous mechanisms through the HLA complex, whether it's, testis antigen, MAGE4, 10, et cetera. Some argue that the persistence is better with TCR and CAR T versus unmodified NK cells. Again, the target repertoire seems to be better with TCR. Tumor specificity, again, better with TCR. But with that said, I'm going to be honest with you that, you have to be careful. Adaptimmune's initial foray into this showed that you can have cross-reactivity of these HLA complexes with other antigens that are expressed on normal tissue like titin. And there is, unlike in unmodified NK cells, really a low risk of, there's really not a risk of GVHD, one could argue, in all of these. But, so I think that some would argue, maybe I'm wrong here, and I'm partly partial, that TCRs at this time seem to be a better overall approach towards solid tumors. But we can debate this, because I know Dr. Yi has some perspectives on it, and I'm sure Marcus also has perspectives on this. Next slide, please. So there are a number of different companies right now in this space. Again, I'm partial because Marcus and I are both involved in Adaptimmune's platform. Obviously, they've sold their asset to GSK and the NYESO, but there are other companies like TerraBio and Cellular and NantWest that are also developing some of these in solid tumors. I think with solid tumor cellular therapy, we're obviously very early in the stage of where compared to where this is in heme. Ironically, I think that we, this space is still very rich and possibly something that will really continue to grow in solid tumors. One thing that I think Andrew wrote in the question was in heme, compared to in cellular therapy in heme compared to the bispecifics, what's going on there? The bispecifics are looking very, very good in heme. They don't look that good in solid tumors. And I will tell you, we've had actually several debates within the CAR-Tox group about, where bispecifics are going to fit in the context of CAR-Ts, and whether even they could even supplant CAR-Ts in heme. And so, even though CAR-Ts and cellular therapy early on seem to be doing great and benefiting in heme, there's a chance that these bispecifics may even supplant that. And where cellular therapies may ultimately be of the best benefit will be in the solid tumor space. Next slide, please. So these are, again, allogeneic assets that are in the clinic. In all disclosure, I'm involved with a lot of the FATE trials, FT500, FT516, and FT536. But there are a number of obviously interesting ongoing and upcoming cellular therapies in solid tumors, and MD Anderson is one of them. We have an alliance with Takeda, and we're developing ongoing solid tumor CAR NK cells with Takeda. Next slide, please. Okay.
Great. Just before we head into the question and answer session, which is of course what this is as well, I sort of want to pose a very broad question, maybe perhaps provocative, but is it too soon to be able to gauge whether eventually allogeneic cell therapy is going to be better than across many metrics or specific metrics versus autologous CAR-T on efficacy, on safety, on logistics? Where does the community lie at this point in terms of what the future outlook is? And I know you touched on versus bispecifics in heme versus solid. Could we maybe expand on which one we expect in another 5 years to really be the leader?
So is the question again, is allogeneic going to eventually supplant autologous therapy, you think, in whether in heme, solid, et cetera? Is that the question?
Yes, and I suppose I'd add on to that, whether we might see allogeneic or autologous niche itself into either heme, either autologous or will they both compete in the same spaces?
No, no. I do think that, I mean, I know that I agree with Cassian and Marcus about 6-month CR and response rates. Those are too early to just kind of make a judgment on apples to apples comparison. But, just looking at the overall landscape, the allogeneic space, at least in heme, looks pretty good. And I just can't see, if ultimately I think these will eventually also get approved, similarly to what we've done with Yescarta and Kymera. And I just think that the marketplace ultimately, unless you do a head-to-head comparison, will in some ways overcome those other products just because of all the logistics that we just talked about. Again, if you were to compare heme versus bispecifics, I don't know the answer to that. Sorry, allogeneic versus bispecifics, I don't know the answer to that. I do think that, one of the things that, advantages of bispecifics is that, these are things that there's still toxicities that you see with cellular therapy, but again, these are things that you can give in the outpatient setting, and I just, these are things that community docs are comfortable, I think will ultimately be comfortable in giving, as long as they learn how to manage some of the adverse events such as CRS and stuff. So we'll see. Doctors are creatures of habit. And we tend to do what we know how to do in the past. And, CD3, CD20 antibodies are not that different than a ADC or a regular monoclonal antibody.
Marcus and I are both Canadians, so very politely let David talk first. I think I just want to mention something really quickly, and I'm less polite because I've been in the U.S. too long. But I think is that the methods that are being tried out now, I think far too early to tell they're effective. What I'm surprised by, actually pleasantly, is that there's some persistence of these transferred T cells, so they're not immediately rejected. Being raised as a bone marrow transplanter, I've always felt like, oh, these guys are just going to not survive at all. But I think that, and maybe it's because some of these patients are slightly immunosuppressed or whatever hand waving we want to call that. But I think also there's so many other different avenues that haven't been investigated to see, is it the CD52 depletion, is and the knockout plus depletion, or is it one of these other maneuvers in which they've been hiding the transferred allogeneic cells? So it's a provocative question. It's way, way too early to answer that. Marcus? Yeah.
So the original question was, is it too early to know whether allo versus auto products are going to be used in 5 years? I think it's too early to know. But you also asked the question, is it from a logistics point of view, allo is clearly the winner. It's just a lot easier to take something off the shelf and use it. From the safety point of view, depends on how the trials go. I think there's long follow-up in auto and allo products, so that's not a difference, although the risk is higher in allo, as we've discussed already in one example. So there's potential for clinical holds to happen because the allo products are being more engineered. And then efficacy, I think it's too early to know, and we'll see. And if your goal is to get something to market, all the auto stuff is getting closer to market now. So that sort of proves that allo, it's not like the allo idea hasn't been around for quite a while, but it's just taking longer, but it ultimately logistically will be so much easier. If it turns out that we only can use auto, is there going to be a mom and pop operation where people have their leukapheresis for their 18th birthday and put their cells on ice for the next 60 years? Maybe that's something that people might decide to do, and then you could make your product later on for auto. But in any case, I think it's too early to know whether allo is going to be the first, or when the timing is going to happen for approvals.
And as the third Canadian, I'll apologize to the audience before launching into the Q&A. But I'll add another question. We didn't go over this in detail, but could we speak a little bit about T cell engagers or NK cell engagers?
You mean like the bispecific or any kind of product that activates T cells? Well, you know, we have talked a little bit about bispecifics versus cell therapies. I think ultimately it's going to depend on durability of responses. The cell therapy, and that's why I don't like the 6-month surrogate endpoint. I want to see something a little bit later. The engagers that require multiple infusions are going to be or patients are going to be less enthusiastic than a one-time treatment that lasts a long time. So if you can achieve long-term control with the cell product, then patients are going to gravitate towards that, and therefore they don't have to keep on getting infusions. One of the dangers of the allo space is that they're seeing rejection of the cell infusions, and the solution is, okay, we're going to give repeated infusions of the cells. What's the difference with a bispecific that's easier to manufacture? So if you end up having to give allo infusions every 2 or 3 weeks, I don't see a difference with a bispecific, and it's probably going to be better to use the bispecific. You know, obviously that's a simplified answer, but it is something. Some of the solutions are making the cells look a little bit like a repetitive drug, and we have to understand what our goals are for the cell therapies.
Thanks very much. And right now we are going to launch into our Q&A session where we will address questions from the audience. And I will call on my colleague, Dr. Andrew Hong, and Vicky Ko, who I believe is on the line in Princeton, to go ahead.
Thanks, Rohan. So I'm curious, Dr. Hong, really fascinating sort of perspective you had around especially sort of looking at cell therapy for solid tumors. I'm wondering, has the time come where one of these antigens is looking to be leading the pack, or as far as selectivity? Because for a long time, there's a question of, you know, how can we realize the vision of cell therapy in solid? The antigen was an issue for a long time. Are you seeing that still being the case, or how is that evolving?
Yeah, I think, can you guys hear me? Yeah, I think that, I think the antigen is still an issue. I do think, and I'm not involved in this trial, even though we did the phase 1 and Marcus and I ran the phase 1 on this trial, but I do think that the SPEAR T cells, they showed close to 40-some% efficacy in the synovial sarcoma space, which is a niche space, albeit. But it's an unmet need, and the phase 2 looks like it's going to be like the phase 1, and we're not going to see a drop in response rates, and the patients are going to seem to have durable responses. I wouldn't be surprised if they get accelerated approval for that, and whether that will be kind of breaking the 4-mile event or not in solid tumors, I don't know the answer to that. But I do think that there are a number of targets down the pike that look good. I mean, I think Marcus and I are also involved in the TCR product. The mesothelin looks pretty good, and that's a little bit bigger population. Logistically, a little bit easier to do in the context that you're not having to screen for HLA, et cetera. So I think that there will be more and more of these kind of targets down the line. I know, and Adaptimmune has already released this, that we're going to be working with Genentech towards towards making these off the shelf with their technology. So, but MAGE-A4 is still a fairly, and they're also developing HLA independent cells. So MAGE-A4 is a small population, but I think that we will figure out other antigens, whether endogenous expressed with HLA or on the cell surface, that may be beneficial in solid tumors. CD70 is something that's I think is interesting. Claudin is another one that's interesting. So I think that we will find additional targets down the line. So I'm a phase 1 doc. I'm an optimist. So I think we're gonna see more and more of these coming down the line.
Sounds good. Thank you.
Can I just add to that a little bit? And I think Davis hit all the high points here of the surface antigens for CAR T-cell based therapies. There are a few more out there. There are some sort of outliers where people have decided, they can make a CAR against a peptide image C molecule and substitute that for a greater affinity. But transitioning to the TCR targets, as we discussed earlier, there are far more targets for TCR, and I think that field has not been mined in a prospective, comprehensive, judicious fashion yet. I think a lot of the lower hanging targets, MYSO, PRAME, MAGE-A4, are being exploited to the fullest extent. Absolutely, in favor of that. I just think that we are ignoring a huge another section. I guess in one of the slides I have is the core of the apple. We've just looked at the peel right now, and I think that that's really going to have to require immunogenetics, proteomics, but also a way to validate these as true targets, not something that's just defined in silico or defined by consensus binding sequences. So that area, there are some companies involved in that, either in shared neoantigen space or shared tumor-associated antigen space. And I can go deeper, but I because of limited time, I just want to make sure that people are aware that there are opportunities out there which haven't been fully addressed at this first layer.
In addition, sorry, Vicky, go ahead.
I just had a question on moving on to unmodified NK cells. So from the talks, it sounds like NK cells are very potent, but they lack persistency. So what would you say is the more important or more clinically relevant parameters here? Persistence or potency? And taking into account that with allogeneic cell therapies as well, there's that potential for redosing.
Potency is a really charged term. So I think we have to be careful how we define potency. And as you know, the most terminally effector cells or differentiated cells, tumor differentiated effector cells are maybe potent from the standpoint that, in vitro, they can kill a target very well because they got tons of perfect enzyme on their surface, or maybe some NFR, SFRs. But I think that potency actually does play into, in general, efficacy when you're talking either of affinity or killing ability, and then that all relates to persistence in a way. So if you were just asking me straight out, do I want a cell that kills better in the plate or lasts longer in a patient, I'd have to go for a cell that lasts longer, but certainly don't want to have to choose. But I think this is an issue that really this is an issue where you like to say, hey, look, I got a more potent cell product, it should work better in the patient, and there is there's a discrepancy that doesn't translate all the time. So I think, an intrinsic property of the cell is most important. Now, since you were talking about NK cells, are there persistent NK cells? Are there memory NK cells? And some work like Kali Malmberg, others Obviously, Louis Lanier's group looking at memory NK cells. Has that reached the clinic? Has that reached a level where you can sort, identify, modify long-term, long-lasting NK cells beyond what Fate is doing with IPS, for example? So the short answer to your question is it's difficult to define potency. Persistence is important in the innate cell. That remains an unanswered question, largely, not completely.
Yeah, I was going to ask you, Cassian. It's like the way you answered that was from a T cell biologist, which you are, perspective, and so it's helpful to have you say that it's basically unknown with NK cells as a therapeutic, whether those rules are going to apply. So I think the future is really interesting to see what happens.
Sort of wondering from a technical point of view, actually speaking to both persistence and quote unquote potency, the idea of CD16 receptors being integrated into NK cells to both increase ADCC, but also prevent antigen escape. Is that something that's catching on, just entering trials, or is it even on the radar?
Depends on the target, right? So there's some people putting this to enhance ADCC against MDSC targets, for example, instead of the tumor target. So yeah, that might be something to consider. I'm just going to answer that question by saying theoretically it's a great idea. I don't know what it does in people, and if it's necessary. Yeah.
Okay, maybe shifting gears for a moment from the technical benefits that we've seen in advancements. Is there anything that you're seeing on the logistical side for streamlining of the actual leukapheresis process, but even for allogeneic, the manufacturing process and the treatment center considerations?
From the standard, I think the fact that I'll speak towards the treatment center, the clinical, the hospitals, even in a environment like Canada, where the government is supporting the healthcare system. The fact that CAR T cells has provided benefit and has resulted in approvals has opened the way for clinical trials to be more easily executed. The feasibility is improved because we have the standard of care CAR T working forward. So investments in leukapheresis centers is happening because you need it for the standard of care CAR, and therefore it's easier to develop new products. And the bone marrow transplanters and the leukemia doctors are working with the solid tumor doctors to take care of patients who are having lymphodepleting chemotherapy. So a lot of barriers have come down from a feasibility point of view, and that's making it much more straightforward in terms of conducting these clinical trials. And there's more buy-in from the institution. So we started with talking about TIL therapy. If you think about how it would have been to institute TIL therapy if CAR T hadn't happened, logistics would be a lot harder. It's a lot of arguing with people about, please bring my patient, let them use some of the heme resources, and that can be a challenge sometimes. So logistically, it's easier, and that's because of the success with the standard of care CAR.
I think something David mentioned earlier was, how we need to have this, facility, as Marcus was saying, whether it flowed from the stem cell transplant, outpatient, inpatient services or not. Can we envision a time when, blood centers might be able to achieve some of this, and certainly not currently. But part of the problem is the preparative regimen and toxicity regimen, and as the three of us know, to some of the stem cell transplant centers, for example, Fred Hutchinson, I can mention specifically, is moving everything to the outpatient because they have the staff to do it as an outpatient and everything else, and follow up, and people live close enough and so on. And but I think that maybe that's an area of ancillary support that's not available to most places, and there's a vacuum for that type of development for not just car toxicity, immunotoxicity, what have you. Yeah.
We hear a lot about consolidation and retreating and enriched dose levels. Is retreatment something that we can expect in the future that will lead to deeper responses, longer responses?
I'm not entirely sure about, you mean the existing products like Kymriah and Yescarta, are you talking about that, or in maybe some future cellular therapy product?
More so, I mean, what we've seen from Allogene and what they've seen with the Alpha trial consolidation regimens, as well as Cellular recently mentioning that the efficacy that they see now, they're hoping to improve by using consolidation therapy, so retreating the patient with the same product.
I think it will depend. I mean, I think there can be a lot of antigen loss associated with the kind of the resistance to these cellular therapies. So you're going to have to double check to see if these patients actually still have the antigen. With CD19, I don't know the answer to that, but I know with a lot of the solid tumor MAGE-A4, for example, the patients who become resistant, and we've really gone back and looked at those, a lot of these patients have antigen loss. I do think that the cellular therapies, as we move forward in the future, and they're already doing this now with the next generation of products, you're going to have more than just CD19, right? It's going to have CD20 and CD19. And likewise, I think the solid tumor products will have multiple antigen targeting products.
So Marcus knows I'm dying to say this. I waited until the last 4 minutes here to say the word antigen spreading, which I think is something we ought to think about, especially since we're conditioning patients with fludarabine and high-dose Cytoxan and so on, that we're eliminating that possibility of endogenous antigen spreading. And so we've shown that others have shown that the importance of antigen spreading, the timing of when it appears, how you promote it and expand it, may obviate some of the requirement for repeated infusions. But I think the next step beyond antigen spreading is effector spreading, with recruiting other effector cells and so on, and how that can be done. So when you ask the question, is multiple infusions required? Obviously, that depends on the T cell product itself, but also what the combinations are going to be in the future.
So following up on the fact that there is antigen escape, just wondering what your thoughts are on combination approaches with cell therapy, for example, adding oncolytic viruses or adding bispecifics. Any thoughts on combination approaches to potentially become the next standard?
Almost in oncology, whenever you see a signal that's promising but not enough, we end up combining with additional agents and having more success with cures. So I think that that'll probably be also the case with cell therapies. As David was pointing out, there's a lot of challenges with the MHC or antigen loss with the TCR-Ts. So if you combine with TCR, say you did a trial with multiple TCRs attacking the tumor, you get MHC loss, but then the T cells are going to be sensitive to NK cell killing. So I think the combination approach is going to be the future. It's whether we can afford it and whether we can make it feasible. And I think that Cassian's point about antigen spreading is a very important one. As a field in general, there's been all this focus on lymphodepletion, and very little for recent in solid tumors and others, looking at ways to enhance the T cell response. All of the NCI work was done with high dose IL-2. We've talked about the problems with high dose IL-2, but definitely an area is how can we make the cells that are infused more effective, either by engineering cytokine receptors so that the cells persist better and are better functioning cells, or giving additional agents to help rev up the response of those T cells. And maybe we can then dial down some of the lymphodepletion requirements.
Thanks. I'm curious, as a follow-up to the idea of multiple antigen targeting, I remember there was discussion about armored or maybe even logic-gated antigen targeting, where perhaps one antigen and another, or perhaps one or another. Can you speak to the degree of maturity that field is in, or is that still sort of more conceptual at the moment?
I think early phase clinical trials is kind of the space where that is. Still early though.
Are you optimistic about that, or is that something that will still has to bear to be seen?
It's overwhelming, all of the different things that are being studied. So, ultimately because venture capital has poured so much money into this space, there's just a ton of stuff happening. And now we need to wait and see what is going to show some interesting results, and then that'll rise to the top, I suspect. That's at least I think it's a bit, there's so much happening. If you look at Asia, all the work happening in China, it's in cell therapies. There's more, you guys are spending your entire careers finding out what's going on, and it's a bit of a needle in the haystack. But, I'm optimistic, but I think time will tell, and we'll have to, conduct carefully controlled clinical trials to really know what's true.
Mm-hmm. Another one. I'm curious if we shift back to the idea of persistence, to Vicky's question about potency or persistence. If we take the persistence angle there and take it one step further, there were a number of technologies we described previously, but I'm curious if you have a favorite, or if there's one that you're most optimistic about for persistence when it comes to specifically allo.
Again, I would put this in the context of neither allo nor auto. If you're talking about allo, then you're going to have to deal with everything else we talked about, in terms of endogenous tumor rejection and so on. I think your starting point is going to have to be intrinsic. Because when you start throwing things, even products out there like Nectar, or what used to be Altor, I guess is now taken over by another company, these sort of additional cytokines, I guess Revimine's got IL-7 and so on. These other cytokines and their products, and now you've got effector specific or non-Treg, non-Treg stimulating IL-2 orthologs and so on. All this is designed to enhance a T cell product that you're kind of going to assume have the receptor on it, but not much more. And I think that's sort of why I sort of believe you need to go intrinsic. You need to see what is the T cell product you're going to put in there, because adding any of these reagents, certainly, in a mouse model or ex vivo in the lab, is going to show expansion of the cells. That's not the issue. The issue is, have you generated a product that epigenetically, intrinsically, what have you, will revert to a memory cell for long-term persistence. And if you're just blasting these things with a cytokine or prolific cytokine, then you're not going to achieve what you think you're going to achieve if you start with the wrong product. So yeah, of course I have a favorite, but that's completely biased. I think it's important to have a genetically modified cell, so it's a memory cell. Now, if you go around selecting cells that are a surface molecule that seems to pertain to memory, you really have to be careful, because large fraction of those cells are irrelevant. And when you put them in, it's only a small fraction that retain memory. Are you going to put in a cell that endogenously, makes, STAT3 signal or STAT1 signal, whatever you have for that. And so I think that's when synthetic biology will come in. There's some products out there. Make sure I don't speak anything out of turn. I think there's synthetic cytokine receptors out there that are important. They're designed to co-opt machinery of the cell in order to enhance persistence. Not to forget, of course, we haven't talked about CD4 T cells too much, but there's a player. Now people have invested a lot of time, effort in for a number of reasons.
Sorry, go ahead, Lydia.
Yeah, following up on your point on how it's better to use something intrinsic, I was just wondering what your thoughts are on the TRuC-T construct that TCR2 Therapeutics is doing, where they use a tumor antigen binder onto this natural, the TCR complex. So they're adopting the natural TCR complex. So any thoughts on whether that would be a safer approach with regards to concerns around toxicity in CRS and ICANN?
I'm not sure if I heard everything you said, but it sounds like they're co-opting most of the TCR machinery, but putting modular fashion the receptor or something on. If I got that right, I'm sorry if I.
Yeah, that's right.
I guess that addresses some of the issues with exhaustion and T cell receptor recycling and all that you like to see. Maybe that the TRAC2 CRISPR technology is designed to address CAR T cells. I guess that's part of the question. It's still the cell that it goes into, and everything else that's associated with memory is sometimes independent of the T cell receptor itself. So you're not going to address all those issues. It's a good start, I suppose. Marcus, do you know if this was the company out of McMaster? Was that? Do you remember? There was another company you reminded me of, Jonathan, our friend.
Yeah, I mean, there's the TRuC point. That strategy is being used by a couple of different companies. There's TCR squared with the mesothelin. There's the Triumvira, I think. David may have experience with that product targeting HER2 new. That comes from Jonathan Branson. So, those trials are happening. David presented the TCR squared data recently. You were asking about toxicity, and that is in vitro or in animals a promising element, but I think David presented that there was some CRS that's been observed. So, it's too early to know what the whether these products are truly going to be safer or not. We'll wait and see.
Fantastic. I think this has been a really compelling and informative overview, but also look forward to what we can expect from the allogeneic, but also solid tumor space, as well as all the considerations for cell therapy overall. I really want to thank the panelists for their wonderful insights. And I think it'll be great to really circle back on this in not too long to see where we are, how the trials are progressing, and whether our opinions have changed. I just want to draw attention to the fact that some exquisite roundtables like the one that we had today are upcoming from Axiom. So we will be having one on precision medicine and another one on disparities in cancer care. We do hope that the audience tunes in for that as well. But again, I really want to give a hearty thank you to our panelists, and I hope to speak with all of you in the near term. Thank you very much.
Right.